Good morning. This is Dr.R.Kamalambigeswari, Associate Professor from Department of Industrial Biotechnology. And today I'm going to discuss on the topic B cell maturation, activation, and its differentiation. First, coming to B cells, they are the B lymphocytes, which are named after their site of origin in the dorsum fabricus in birds and in bone marrow in case of humans. They form the basis for humoral immunity by the production of antibodies or immunoglobulins. And these B cells are responsible for giving adaptive immunity, and they are very antigen specific. They have high antigen specificity, and they are responsible for raising humoral immunity, which is mediated through the production of antibodies. And the final destination of B cell is plasma cell. That means the B cells will differentiate into plasma cell, which are responsible for the production of antibodies. This is regarding the cellular and humoral immunity. The lymphoid progenitor cell will be having its transformation either as a T lymphocyte or as a B lymphocyte. Once the B lymphocytes are produced, then of course they will differentiate into plasma cells, which are responsible for humoral immunity. And once they have been transformed as T lymphocyte in thymus, then of course they are responsible for cellular immunity, that is cell-mediated immunity. And coming to B lymphocytes, once it encounters the antigens with the help of certain cytokines, it has the ability to go for clonal expansion, and it has the ability to get expanded or differentiated as memory B cell and plasma cell. And this plasma cell are responsible for the production of antibodies or immunoglobulins, which in turn will give humoral immunity to protect us from various antigens. Coming to the development of B cell, the differentiation of B cells occurs in the bone marrow throughout the life of an individual. The earliest recognizable cell in the B cell lineage is pro B cell, which is followed by pre B cell. That means the precursor for this pre B cell is pro B cell. And this pre B cell will develop into an immature B cell. And the immature B cells are those cells which express the IgM on its surface. It's nothing but immunoglobulin M. The immature B cell are characterized by the expression of this immunoglobulin alone, that means this IgM on the surface of the cell. And the mature B cell will be obtained from immature B cell, and this mature B cell will have the ability to express two different types of antibodies on the surface, like IgM and IgD. This regarding the development of B cell, the lymphoid stem cell has the ability to get transformed to pro B cell, and from this pro B cell, the pre B cell arises, and this pre B cell gives rise to the immature B cell, and the immature B cell gives rise to mature B cell, and the mature B cell gets activated due to the encounter of antigen, and the activated B cell has the ability to differentiate into two different types of cells like plasma cell and memory B cell. Both are responsible for providing humoral immunity. And this immature B cell will express only IgM on the surface, whereas the mature B cell are characterized by the expression of two different types of immunoglobulin like IgM and IgD. Here, the antigen independent phase occurs in the bone marrow, and the antigen dependent phase occurs in the peripheral lymphoid organ. Next is about the activation of B cell. And the activation of B cell leads to the proliferation and differentiation of B cell to plasma cell, which is the end stage of B cell differentiation. It occurs in specialized regions of secondary lymphoid organs like the spleen, the lymph nodes, and the germinal centers, where the B cell is going to encounter the antigen. And the B cell gets activated by two different means. One is through the encounter of antigen, next is through class or isotype switching, which is nothing but the production of various different types of antibodies. Here, we'll have the representation where the B cell is going to get differentiated to the plasma cell. Various antigens, cytokines, and through the T cell signaling, the naive B cell, that means which does not encounter an antigen, will get activated. And once the B cell activated, it is going for differentiation to plasma cell. Whereas the unactivated B cells will die. And coming to T cells, the T cells will be characterized by the presence of T cell receptor, and this T cells will also be activated on encounter of antigen as well as by co-stimulation signals like cytokine signaling and all. And the T cells will also be activated, and those T cells which have not been activated will also die. And the T cells, once it has been activated, it is going to release various cytokines which are necessary for uh, which where the cytokine is going to act as a signaling molecule for the immune system. Here, the immature B cell has been uh, uh, developed as a mature B lymphocyte, where it's going to express two different types of antibodies like IgM and IgD. And these two different types of antibodies, which is present on the mature B cell, will be going for a class switching, where which has also been um, uh, allowed to receive the various cytokine signaling like interferon gamma, interleukin 5, and interleukin 4. Once mature B cell receives the interferon gamma signaling as well as with the help of Th1 cell signaling, it is going to produce the immunoglobulin IgG. Thereby, it is going to act as a plasma cell which is producing the immunoglobulin G. And once a mature B lymphocyte is going to receive the signaling like interleukin 5 signaling and Th2 signaling, then of course it is going to be differentiated as a plasma cell, which is going to produce the immunoglobulin A. And a mature B lymphocyte, once it receives the signal like interleukin 4, as well as with the help of Th2 cells, by the interaction with the Th2 cells, it is going to be differentiated as a plasma cell, which is going to produce IgD. So all this comes under the class switching. Now let's see about the function of B cells. Mainly, the B cells are uh, involved to interact with the antigenic epitopes using their immunoglobulin receptors. So on the surface of the B cell, there will be the antibodies like immunoglobulins, and these immunoglobulins will interact with the antigenic peptides. Thereby, it has the ability to induce the humoral immune response in the individual to encounter the antigen and to present antigenic peptides to T cells, which consequent upon the internalization and processing of the original antigen. Sometimes the B cells may also act as an antigen presenting cell, where it's going to process the antigen, and the processed antigen will be received by the T cells through the MSC molecules, and so that the T cell will go for activation, and it will also release cytokines, which is going to act as one of the signaling molecule in the immune system. And these B cells are also responsible for a humoral arm of the adaptive immune system, which is going to act against many intracellular pathogens as well as extracellular pathogens. The immunoglobulin, once when bound to the cognate antigens, it can activate the whole complement system and help the phagocytes to take up the antigen. These are the various stages of B cell development. The B cell actually arises from the stem cells, where we call as hematopoietic stem cell. And this hematopoietic stem cell may get transformed to be a pro B cell, and the pro B cell develops as a pre B cell. And from pre B cell, the immature B cell arises. And from immature B cell, the mature B cell arises. And each stage of this B cell development is characterized by various distinct cell surface markers, as well as by the specific pattern of immunoglobulin gene expression. The immunoglobulin gene rearrangement, which takes place in B cell, is responsible for the production of antibodies by these B cells. So here we have the stem cell, which indicates the stages of maturation. The stem cells get transformed to be a pro-lymphocyte, and from this pro-lymphocyte, we get pre-lymphocyte. And this pre-lymphocyte, in case of pre-lymphocyte, the immunoglobulin molecule starts to express, but it is not fully functional. And then the pre-lymphocyte will get transformed, or it get developed as a immature lymphocyte, where IgM alone will be expressed, and then the immature lymphocyte will get developed to be a mature lymphocyte, and the mature lymphocyte goes for differentiation and it produces various effector cells. Coming to the pro B cell, it is one of the earliest recognizable B cell stage, and it has the following surface markers like CD45, Ig alpha, and Ig beta heterodimer, which are the part of B cell membrane receptor, which is in the later stage. And of course, it is characterized by the presence of CD19, CD24. It is another cell surface marker of pro B cell, and next it has the marker like CD43 as well as c-kit. So this c-kit is one of the uh, cell surface marker of important cell surface marker of B cell. And the heavy chain gene rearrangement begins in the stage, that means in the pro B cell stage itself, and its completion will be at the end of the stage. The pre-B cell is characterized by the beginning of the translation of heavy chain gene, and here it takes place essentially in the bone marrow stromal cell. The bone marrow stromal cell, it is one of the cell which is going to serve as a platform for the development of B cell. And the mu chain is usually first rearranged, and the light chains are not synthesized at the stage, and the small peptide called surrogate light chains are synthesized, but this is not the true light chain. This is regarding the production of antibodies, and they complex with this mu chain, and this complex will be expressed on the cell surface. This is characterized at the pre-B cell stage itself, and the mu chain and the surrogate light chain complex associate with the Ig alpha and Ig beta heterodimer to form a pre-B cell receptor. So here at the platform, we could find the stromal cell, and this stromal cell will provide the nutrients that is essential for the B cell development. So the pre-B cell arises from the late pro B cell, and the late pro B cell arises from the early pro B cell, and the pre B cell now it has the ability to secrete cytokines like interleukin 7. The immature B cell, it begins with the light chain gene, and only one type of light chain is expressed, either kappa or lambda, and the light chain associates with the new chain in case of this immature B cell, and hence a true IgM expression occurs on the B cell surface. So the immature B cell is characterized by the presence of immunoglobulin M. This also results in B cell committed to one particular antigenic specificity as determined by the binding site of surface IgM. This is regarding the mature B cell. The mature B cell is characterized by the expression of two different types of immunoglobulin like IgM and IgD on the presence of or on the cell surface. And the mature B cell, once the B cell gets mature, it leaves the bone marrow and it enters the peripheral lymphoid tissue where they get activated on encountering the antigen, and it produces two different types of effector cells like plasma cells and memory cells. The plasma cells are short-lived cells. They have the ability to produce antibodies, whereas the memory cells produce second immune response after they re-encounter the same antigen. So this memory cell are long-lived cells, and they are responsible for lifelong memory of antigens. If the memory B cell cannot encounter the antigen, of course, they die in the bone marrow itself within few days by means of the process called apoptosis, which is said to be programmed cell death. Here we could find two different stages of B cell development, like in maturation, which is going to get explained as antigen independent phase, and then the activation and differentiation of B cell, which is the antigen dependent phase. So the pro B cell, which is characterized by the surface marker like CD45R, is going for the development, and it is going to get developed as a mature B cell. So during the stage of its development, the immunoglobulin gene rearrangement takes place, as well as the selection process occur. Thereby, it is going to get developed as a mature B cell expressing two different types of antibodies like IgM and IgD. So once a B cell maturation occurs in the bone marrow, the B cell leaves the bone marrow and enters the blood. And from the blood, it enters the various peripheral lymphoid organs. So in the peripheral lymphoid organs, it goes for antigen-dependent phase of activation and differentiation. The naive B cell, which has not encountered the antigen, will encounter the antigen once it enters inside the peripheral lymphoid organ like spleen, lymph node, and all. And those naive B cells, which have not encountered the antigen, will go for cell death. About 90% of the B cells will not encounter the antigen, though they are said to be the mature B lymphocyte. Those lymphocytes, even after entering the peripheral lymphoid organ, will not encounter the antigen, and those cells that have not encountered the antigen will go for apoptosis, which results in cell death. And those B cells that has been activated by encounter of antigen as well as by receiving the stimulus from TH cell will be activated and will be differentiated to two different types of B cells like plasma cells and memory B cells. The plasma cells are responsible for the secretion of antibodies, and memory B cells are responsible for lifelong memory of antigens. And this slide will explain about the activation of B cells, which is going to occur in the peripheral lymphoid organ. It's an antigen-driven mechanism. Once B cells encounter the antigen, it will get activated, and the activated B cell will go for clonal selection. And finally, it results in two different types of cells like plasma cells and memory B cells. And in the absence of antigen-induced activation, the naive B cells will die within few weeks by apoptosis. Here we could see the lymph node where the uh, antigen has been encountered by the B cell, especially on the cortex of the lymph node, which is said to be the primary follicle. So once the B cell that is present in the primary follicle encountered the antigen, the primary follicle turns into a secondary follicle, and there it encompasses the germinal center. So there are various changes that occurs in the germinal center, like somatic hypermutation, selection, and class switching. All is necessary for the differentiation of B cells. So this is regarding the somatic hypermutation which takes place in the B cells. So while the somatic hypermutation takes place in the B cell, then it induces the B cell to undergo rapid cell division. And here the mutation of genes occurs randomly in the B cell, and the mutation of genes may increase the antigen binding affinity of membrane immunoglobulin that are found on the B cell, or it may decrease the antigen binding affinity of membrane bound immunoglobulin on B cell, or sometimes this somatic hypermutation may be silent. That means it may not increase or decrease the affinity of antigen binding, a characteristic feature of membrane bound immunoglobulin on B cell. So this is regarding the selection process. Selection of selection of B cells in the sense those cells that has been activated by antigen. So here the centrocytes compete with each other to bind with antigen on follicular dendritic cells, and those B cells which bears the high affinity membrane bound immunoglobulin are most likely to compete, and those that fail will die by apoptosis. And signal will also be received from T helper cells for further survival of this B cell. And once the B cells uh, bind with the antigen and also receives from the receive signal from T helper cells, they will differentiate into memory B cells or plasma cells. So this is regarding the clonal selection of B cells. So the B cells that has various antigenic specificity will encounter the antigen molecule based on the affinity with antigens, and here, the selected B cells will proliferate, forming a clone of identical cells bearing the same receptor for the specific antigen, and then they differentiate into a clone of plasma cells, or else they may differentiate into a clone of memory cells. And some proliferating cells develop into short-lived plasma cells that secretes the antibodies which are specific for the antigen. And some proliferating cells develop into long-lived memory cells that can respond to subsequent exposure of the same antigen. So this is regarding the class switching or isotype switching. So here the B cell changes its class of immunoglobulin that is produced while preserving the antigenic specificity. It allows the VH domain to associate with the constant region of any isotope of the antibody. So that enables the antibody specificity to remain constant while the biological effector activities of the molecule vary, which depends upon the constant region. So the interaction between CD40 on the B cell and CD40 ligand on the TH cell, CD40L on the TH cell is essential for the class switching activity of B cell. It occurs only during active immune response. So this is regarding the activation of B cell. The B cell is going to be activated with the help of various cytokines like interleukin 2, 4, and 5. And the proliferating B cells will be once they are exposed to interferon gamma and tumor growth factor beta, they have the ability to get differentiated as the plasma cell, producing IgG2 or IgG3 type of plasma cell, and subsequently IgA or IgG2B type of plasma cell. That means the plasma cell has the ability to produce these different types of antibodies. And if the interleukin 4, if the proliferating B cells receives the interleukin 4 signaling, then of course it results in the differentiation of plasma cell, which has the ability to secrete IgE or IgG1. Likewise, if the proliferating B cell receives the interleukin 2, 4, and 5, then of course it has the ability to differentiate into plasma cell, which secretes IgM. Coming to plasma cells, these are the membrane bound. The plasma cells contain the membrane form of immunoglobulin, which changes to secreted form, and the rate of transcription of heavy and light chain genes here significantly varies. Coming to this memory B cell, they are seen in T-dependent immune responses. And these memory cells are responsible for long period activity. That means they'll remain in the blood for a longer time, and they are responsible for lifelong immunity. And they have high affinity antigenic receptors, and they are capable of mounting rapid responses to subsequent introduction of antigen of the same antigen. And they remain in the lymphoid organ where they are generated or else they will be recirculated. Now coming to the immunoglobulin, this is a immunoglobulin or the antibodies, which is a glycoprotein that is made in response to an antigen and can recognize and bind to the antigen that causes its production. Coming to the structure of immunoglobulin, they are composed of 4 polypeptide chains, 2 identical light chains, and 2 identical heavy chains. And these 2 identical light and heavy chains are linked by disulfide bonds. And the light chains or they are similar in all immunoglobulins. And the light chains occur as 2 different types, either kappa light chain and lambda light chain. And the light and heavy chains are subdivided into variable as well as the constant regions. And each heavy and light chain contains amino terminal in variable region and carboxy terminal in the constant region that we can infer from the structure of this immunoglobulin. The heavy chains are structurally and antigenically distinct for each and every class of immunoglobulin like IgM, IgA, IgD, IgE, and IgG also. Here, each immunoglobulin peptide chain has intra-chain disulfide bonds, which forms as a loop that we can infer from the structure of the immunoglobulin, which is shown here. And each loop is compactly folded to form a globular structure domain. So here we have the variable domain and the constant domain in case of light chain as well as in case of heavy chain. So the light chain contains a single variable domain VL, which is designated as VL, and a constant single constant domain, which is designated as CL. Whereas the heavy chain contains one variable domain VH and a three constant domain CH1, CH2, and CH3. The hinge region is the segment in heavy chain between CH1 and CH2. So while the immunoglobulin is digested with the proteolytic enzyme, especially papain, then it produces three fragments, two identical FAB fragments, which stands for fragment of antigen binding activity, and the other fragment called FC fragment, fragment of crystallization. So here the FAB fragment are responsible for binding with antigen, and the FC fragment, which is said to be the fragment of crystallization, is responsible for exerting the biological activity of immunoglobulin. So once the antibody immunoglobulin is digested with pepsin, then of course it produces a single fragment which is composed of two FAB fragments like subunits, FAB2 kinds antigen, and FC fragment which is not recovered and it is digested to small numerous peptides. So once the antibody is subjected to pepsin digestion, then of course FAB fragment and the FC fragment, a single FAB fragment and a single FC fragment can be obtained, but this FC fragment will not be recovered as a whole. It can be recovered as small peptides. That means it is digested a lot. Coming to the classification of immunoglobulin based on the structure and based on the antigenic nature of heavy chain, the immunoglobulins are classified into five classes like IgG, IgA, IgM, IgD, and IgE. Coming to immunoglobulin G, it is one of the abundant classes that is found in serum. It constitutes about 80% of total immunoglobulin, and it is characterized by the presence in blood, plasma, as well as in tissue fluids, and it contains less carbohydrates than other immunoglobulins, and it has a half-life of about 23 days, which is the longest of all the immunoglobulin isotypes. And this immunoglobulin G crosses the placenta and it provides natural immunity to fetus as well as for the neonates during their birth. And it acts against bacteria as well as viruses by opsonizing it. That means they'll coat on this antigen and they'll allow this antigen to get opsonized by many antigen presenting cells. And these antibodies will also neutralize the toxins and they will activate the complement by classical pathway. And they are responsible for the catabolism of various toxins also. And the catabolism of IgG is unique in that it varies with its serum concentration. We have various subtypes of this IgG, which is said to be the subclasses of IgG, like IgG1, IgG2, IgG3, and IgG4, which is characterized by the variation in length of hinge region as well as by the presence of disulfide bonds. Next, we'll see about the major histocompatibility complex, which is called as MHC. So this major histocompatibility complex is a set of surface proteins which are located on the cell membrane of all nucleated cells. In humans, it is called as HLA, which stands for human leukocyte antigen. And this major histocompatibility complex plays more important work to identify the antigen between self and non-self body, which is highly essential to avoid many autoimmune diseases. And this is responsible for intracellular recognition as well as it is responsible for antigen presentation. And histo here refers to tissue, compatibility refers to living together harmoniously. So MSC molecules, they will always recognize only the T lymphocytes, and the two types of MSC are worked in immunity. like T helper cells, which are recognized by MSC molecules 2, and cytotoxic T cells, which are always recognized by MSC molecules, MSC 1 molecule. And this major histocompatibility complex is membrane attached protein, which work on regulation of antigen between self and non-self body, as well as it's responsible and it is involved in antigen presentation. There are different types of MSC molecules like class 1, class 2, class 3, and class 4, which plays its innate role in immunity. Coming to class 1 MSC molecule, they are characterized by the presence of by their molecular weight, like they'll have the molecular weight of about 45 kilodalton, and they form the major group of MSC molecule. They are present on the surface of all nucleated cells except the nervous tissue as well as platelets. And it presents the antigen to T cells, T cells especially, and it binds with CD8 adhesion molecules of TC cells, and it brings about the cell-mediated immune response on interacting with TC cells. Coming to the structure of this class 1 MSC molecule, it consists of two polypeptide chain, namely alpha chain and beta 2 microglobulin. The alpha chain, which is non-covalently attached with the beta 2 microglobulin, here the alpha chain contains a transmembrane glycoprotein, which is encoded by the A, B, and C gene of good HLA. Here, the alpha chain is characterized by the presence of three domains like alpha 1, alpha 2, and alpha 3, and each domain consists of about 90 amino acid sequences. And this beta 2 microglobulin, which is non covalently attached to the alpha chain, is similar in size and it does not contain the transmembrane protein. And once the antigen is internalized, it is processed and inside the proteasome, and the peptides are produced. And these peptides are further loaded on the groove of MSC1 from the endoplasmic reticulum. So here we could see the structure of a class 1 MSC molecule. So here the alpha chain is present and the beta 2 microglobulin is non-covalently attached to alpha chain. And the alpha chain consists of three domains like alpha 1, alpha 2, and alpha 3. And here we have a membrane spanning domain of this class 1 MSC molecule, which pierces the or which travels via the cell membrane of the antigen presenting cells, and the cytoplasmic tail is present in the cytoplasm of the cell. Coming to the structure of class 2 MSC molecule, they are present on the surface of antigen presenting cells, and the cells which engulf the foreign antigen, and it binds with the exogenous antigen, which is presented via or which is processed via the endocytic degradation, and it binds with CD4 adhesion molecules of TH cells, that means T helper cells, and it also consists of two polypeptide chains, namely alpha chain and beta chain, and the antigen is processed inside the endosome, and the peptide is further loaded on the groove of class 2 MSC molecule. So here we could find the structure description of class 2 MSC molecule. The class 2 MSC molecule consists of two polypeptide chains, namely the alpha chain, which is of 33 kilodalton, and the beta chain, which is of 28 kilodalton. And the both chain are attached non-covalently, and each chain contains two units. The two units of alpha chain are called alpha 1 and alpha 2, and the two domains of beta chain, they are called as beta 1 and beta 2. And beta 2 and alpha 2 are transmembrane domain anchoring the MSC2 plasma membrane. The alpha 1 and beta 1 domains jointly bear a peptide binding group. So here we could see the structure of class 2 MSC molecule. We have two chains like alpha chain and beta chain running parallelly, and we have the transmembrane segment also. Here we could find the peptide binding group for attachment of the antigenic peptide, and here also the membrane transmembrane segment is present, and the cytoplasmic tail is present for class 2 MSC molecule. Coming to the class 3 MSC molecule, the molecule includes complements like C2 and C4. And coming to class 4 MSC molecule, these molecules is present on the T cells of leukemia, as well as it is present on many immature thymocytes.